Our lab at the Technion Faculty of Biomedical Engineering is focused on stem cells and tissue engineering for regenerative medicine. We engineer tissue construct like bone, skeletal muscle, cardiac tissue, spinal cord tissue, and the idea is to engineer tissue that can really fully integrate after implantation and become functional. We focus on cell-cell interactions, various multicellular seeding, and also the interaction between the cells and the biomaterials.
People from all over the world, the patients, physicians, biotech companies and food companies reach out to us with their specific problems, and we come up with tissue engineered solutions.
We decided to do donation that this time will have a significant impact on humanity, things that really can help people in specific areas. One of the areas was diabetes, and we started the search to find somebody that can help us, not just to find mechanism to monitor, etc., but to actually to cure, which was very, very different than everything else that was done before.
The main goal of type 2 diabetes treatment is to normalize glycemia and minimize chronic diabetes-related complications. Current treatment approaches consist of lifestyle modification and oral and injectable pharmaceutical agents. While insulin resistance at the peripheral tissue level is the key feature of type 2 diabetes, except to seldom used PPAR gamma activator, none of the commonly used drugs directly address it. Skeletal muscle is responsible for up to 80% of glucose uptake in response to insulin produced in the body. Adipose and skeletal muscle tissue express an insulin-stimulated glucose transporter called GLUT4. When insulin binds to its receptor, the glucose transporters translocate to the plasma membrane and permit glucose entrance into the cells. However, in diabetic patients, concomitantly with impaired signaling, cellular GLUT4 expression is reduced and its translocation is impaired, which results in reduced insulin action.
The solution that we came up with is the engineered skeletal muscle enriched in GLUT4. This construct can be used as an actual treatment in diabetic patients.
So in order to create the engineered constructs, we took skeletal muscle precursor cells, the satellite cells, and genetically modified them to overexpress the GLUT4. And we seeded them on 3D biodegradable scaffolds and cultured them in order for them to develop into a mature skeletal muscle tissue. And these engineered constructs, they had a 4-fold increase in glucose uptake upon insulin stimulation. Following these results, we implanted the engineered constructs in the abdominal muscle of diabetic mice, and the constructs integrated fully into the host tissue. And we followed the fasting basal glucose levels throughout the 4 months, a long period in mice. And the constructs had an impact on the whole body metabolism and whole body glucose levels. There was a normalization and a reduction of 26% in the glucose levels in the blood, and also we saw a beneficial effect on other aspects of the metabolism in the body. We saw a reduction in fatty liver symptoms, and also we saw an increase in myokines that have a positive effect on the diabetic state.
So to bring this technology closer to clinical use, we're currently developing a new approach for minimally invasive delivery of the skeletal muscle tissue. We are developing constructs that will be injected straight into the muscle, thus avoiding the pain, the risks, and the discomfort associated with an open surgery.
This is going to really change that whole notion of how you deal with diabetes, because it's not maintaining it, it's really overcoming it.
Our new therapeutic tool has the potential to restore insulin sensitivity in diabetic patients and also to maintain healthy blood glucose level over a prolonged period of time. Moreover, this engineered muscle tissue construct has the potential not only in diabetic patients but also to repair other muscle pathologies. Yet we have to keep in mind that everything presented here is still in basic research.
We are confident that all of you share our vision that the studies we presented here will soon mature into innovative and effective therapeutic options for diabetic patients. Our team promises to move forward to achieve this goal.